Sebastian. I'm in the Department of Immunology at Duke University. I'm a first-year PhD student in the lab of Ashley Mosman. And I'd like to talk to you today about immune barriers of the central nervous system. So how our brain defends itself from pathogens and what mechanisms our body has to regulate inflammation and defend itself from the outside world. So the immune system is built to determine self from non-self and try to keep out pathogens from our body. And it does a really good job of this. The immune system has specialized mechanisms to keep out parasites, bacteria, viruses, pretty much anything that the world can throw at it. But granted, it's not perfect in doing this. Sometimes when the immune system gets out of control, it can recognize itself as a foreign thing, and that leads to autoimmune diseases like type 1 diabetes, lupus, inflammatory bowel disease, or chronic inflammation can occur where it overreacts to a threat, and that leads to things like asthma, psoriasis, and a variety of other diseases. So it's in this constant balance where it wants to be aggressive in order to fight off pathogens, but also restrain so as to not cause damage to the self. And how does this work in the brain exactly? So obviously, the brain's a really important organ. Most of us use it every day, not me sometimes, but I think it's really important to try to understand how the immune system might work here, because you can imagine that if it did become overreactive or cause inflammation, that would be absolutely catastrophic to the way that our brain works. And it could lead to death or any kind of disability. So for a long time, it was thought that basically the immune system stayed out of the brain. It was what immunologists called an immune privileged organ. It just didn't go there. And there was evidence to back this up. So if you were to do a transplant on a normal part of your body, like the skin, you're from a foreign subject to your own, you would get a rejection. Your body would reject that graft completely. However, if you transplant something into the brain, it stays there, and that's an indication that the immune system isn't really reaching its way in there and rejecting the graft. They were also kind of unable to find any typical immune cells in the brain. So for these reasons, they came up with the idea of immune privilege, and it was just determined that there wasn't an immune system in the brain. But recently, there's been a lot of experiments done that kind of flip that narrative on its head, and I want to talk to you about that today, as well as some of my own research. So people weren't stupid when they were coming up with this idea of immune privilege. They knew that the brain still had to be protected from pathogens. And some of the ways they thought this occurred were with physical barriers. So your skull is really, really thick. It's hard to punch your way through that. Don't try, but trust me, it's really hard. In addition to the skull, you also have the blood-brain barrier, which prevents any outside material from traveling from the blood into your brain. This is a really tight junction that surrounds the blood vessels in the brain and only selectively allows certain agents to get through. This is one of the reasons the brain is notoriously hard to get drugs into, because our body does a really good job of forming tight junctions to block that out. However, recent advances show that these aren't the only barriers to the brain and that the immune system does play a role. So here I've shown in the green on this side, that's a slice of a mouse brain, and in the green are cells called microglia. And these are resident immune cells that live in the brain. You might know already that the brain is made primarily of two types of cells: neurons, which relay information, and glial cells, which support them in that process. Microglial cells fall under the glial category, but they're actually of their own distinct origin and come from immune progenitors. So they serve a variety of functions in the brain, with basic tissue homeostasis, helping to form new neuronal growth. They help clear up debris that's accumulated in the brain, and they also provide a last line of defense against pathogens. In addition to the actual parenchyma of the brain is surrounded by 3 layers of membranes called the meninges. And a few years ago, it was discovered that within the meninges, there are lymphatic vessels. This is important because lymphatic vessels allow for immune cells to travel throughout the body, so they can respond to infections in the brain and infiltrate it, and they can also report back to the rest of the body to try to initiate immune responses. It's actually funny, so this was discovered 4 years ago now, but actually there was evidence that in the late 1800s, people had discovered these lymphatic vessels during dissections, but it was lost to history. So it just goes to show that we discover and rediscover things all the time. But This, as well as the discovery of microglia, shows that there are immune barriers around the central nervous system. However, despite these barriers, infections can still impact the brain. And here's a list of pathogens that do infect the brain, and they cause tens of thousands of deaths every year in the United States. So you notice some fairly common ones like influenza, E. coli, but a variety of pathogens can make their way to the brain: bacterial, viral, fungal, parasites. And two that I want to call your attention to that my lab works with are Naegleria fowleri and VSV, which is a model of rabies and also influenza to some degree. So we use those two pathogens in order to study immune responses at the central nervous system. Now, in addition to pathogens that can affect the brain, the immune system is not perfect as far as inflammation and autoimmunity goes as well. So if you remember that balance from the beginning, there are autoimmune diseases of the brain. So multiple sclerosis is an example of one where immune cells recognize the myelin sheaths that surround your axons and eventually degrade them, leading to debilitating disease. Another really promising area of research right now is that the immune system might be involved in the pathogenesis of Alzheimer's. So it's believed that perhaps defective microglia aren't able to clear out amyloid beta plaques or other debris in the brain, which could lead to Alzheimer's, or that inflammation coming from immune cells in the brain is contributing to the pathogenesis of neurodegenerative diseases. So the immune system's not perfect, either at defending pathogens in the brain or preventing autoimmunity. So the goal of our research is to study the immune system, understand how it works, and perhaps improve it to therapeutically treat either of these types of conditions. So you may have heard before in the news of Naegleria fowleri, which is a deadly brain-eating amoeba. It always generates really big headlines whenever there's a case. It's common in the southern United States. People get it from swimming in lakes, or if they're trying to treat themselves with teapots that they pour up their nose, but they don't boil the water correctly. People can die of this disease. It's fairly uncommon. Only a few hundred cases have been reported in the US, but it's 99% lethal, extremely deadly, and it kills whoever is infected within 12 days. It works very, very rapidly to eat its way through the brain. So how is this able to get past the immune barriers that exist within the meninges, past the microglia, past all the physical barriers, and infect someone? Well, we use this as a model organism to try to understand that. And it turns out that there are actually a couple of chinks in the armor of your immune defense in the brain. One of those is the olfactory epithelium. So the olfactory system is unique because it represents a direct gateway from the outside world into your brain. There are only a couple of instances that allow pathways into the brain. This is the only example that allows a direct portal from the environment to your brain. So the olfactory system works by having these neurons in your nose that sense odors, and they travel along long tracts into the olfactory bulb of the brain. They represent a single-cell gateway from the outside world to the brain to relay the sense of odor to your brain so you can make sense of the world around you. So this represents a very unique pathway for pathogens to potentially exploit and invade the brain. This is how Naegleria fowleri and a few other pathogens work to infect the brain. So we study this in order to understand the immune barriers that exist here and try to seal them off to pathogens. Now, I was taught growing up that you can pick your nose and you can pick your friends, but you can't pick your friend's nose. So we don't study this in humans. We use mice as a model in order to understand what's going on here. People don't really like it if you're jamming things up your nose and trying to pick out, all their boogers to study. But the mouse has a similar olfactory system to humans, although theirs is a little bit more complex. They're much better at smelling than we are. And you'll notice that these convoluted structures here, these bony turbinates, are much more complicated than the simple structures in the human nose. But on these turbinates, there exists an epithelial layer of cells that are laden with olfactory sensory neurons, these neurons that are sensing odor and traveling back to the brain. So you can see in that bottom right image how they have cilia that extend into the mucus, sense odors, and travel down through the epithelial layer back towards the brain. And there are a variety of cells that exist alongside these neurons to support them and help create a barrier. What I'd like to talk to you about today are some of the immune cells that exist within this epithelial layer that help form a protective barrier against all these pathogens that we're encountering by inhaling every single day. And you can see in this picture here on the left that those olfactory sensory neurons are in green, and you can tell that they're really, really densely layered throughout this epithelium. And you can see those circles, which represent the axons traveling back in towards the brain. So this is a similar picture, but rather than the olfactory sensory neurons being labeled, in green now are macrophages, which are a type of immune cell that are closely related to the microglia in the brain. And they're called macrophages because macro means big and phage means eater. They're really good at gobbling up debris, gobbling up pathogens, and destroying them. So they represent the first line of defense against pathogens in this epithelial layer. If you were to zoom in, you can see that they're extending these long finger-like protrusions into the epithelial layer as they try to sense pathogens and monitor them. They're also really important in maintaining tissue homeostasis. So in green here are those olfactory sensory neurons as they travel back towards the brain. And in red are immune cells. And you can see how they directly interact with those neurons. They're scanning for pathogens, they're trimming them and maintaining them, and they represent a really important part of this biology in defense against pathogens. But sometimes macrophages aren't enough. If you were infected with something like Neutrophils fowleri or a virus like DSV, you would have to call in a lot of other help. And macrophages are really good at signaling to other cells to try to get them to come out when they're presented with a challenge. So in red here are a type of cell called neutrophils. And neutrophils are basically the kamikaze pilots of the immune system. They basically exist to rush to the site of infection, come out of the bloodstream, and blow up. And they will throw up their DNA over pathogens to try to coat it, block it, do anything they can to get it from invading the central nervous system. So you can see here, the blue is kind of labeling all those turbinates of the olfactory epithelium, and the red cells, the neutrophils, have actually thrown themselves out into the airway. So they're literally extruding themselves from the body into the air in order to fight these pathogens. So this is an example of VSV infection. And you can see how much damage the infection has caused anyway, despite the neutrophils. A lot of the epithelial layer has been stripped off of the turbinates, and it's caused a lot of damage. Neutrophils can also come into the brain. So this is the olfactory bulb of the brain. And if you notice, there's a lot of holes in it. And those holes are caused by the brain-eating amoeba Neutrophils fowleri. And the neutrophils have kind of swarmed to those holes to try to contain it or stop the pathogenesis of that pathogen. And this could either be good or bad. They could be slowing it down, or they could be causing even more inflammation that's causing a lot of damage in the brain. And ultimately, they aren't able to completely contain it. Mice will normally die on this infection. But the immune system can eventually mount an effective response to pathogens in the olfactory epithelium and keep the brain safe. So if a mouse survives its first viral infection and is re-challenged with the same virus, it now has T cells located within the tissue that can fight it off immediately and prevent reinfection. So the T cells here are another type of immune cell, part of the adaptive arm, that come in with specific memory against the pathogen that the organism was initially challenged with. And they're residing in the tissue and waiting. When they're re-challenged by the red virus, you can see they start to release a molecule called interferon gamma in green. And by doing this, they're able to prevent the virus from really taking hold, and the mice survives easily. So part of our research goal is to understand, how can we initiate this adaptive response through vaccination? How can we speed up this, how long it takes for the T cells to get there? If we can get them to respond more rapidly, perhaps we can prevent Neutrophils fowleri and other viral infections from infecting the brain. So I wanted to leave you with a little video, which I don't know if it's going to work in here, but I'm going to try pulling it up. So using a technique called intravital imaging, we're able to go inside the brains of live mice and see what's going on. In this teal color, we've labeled both neutrophils and macrophages, and you can see how they're responding to a Neutrophils fowleri infection. So you see several little blobs here of cells that have surrounded where the amoeba is and are trying to get it to go away. You can even see one kind of as the amoeba is moving down through the brain, how the cells will follow it. And in addition to these cells that are swarming and surrounding it, you also see some that are traveling through the blood vessels and trying to report to the rest of the immune system to get a response to occur. So using these techniques, we hope to understand exactly what's going on during the course of these infections and understand how we can better prime the immune response in order to fight them off. So thanks for having me here today, and I'm happy to take any questions. I'd also like to thank my lab, Ashley Mosman, who's the PI at Duke, and his wife, Annie Mosman, who also works in our lab. Thanks.